Okay. We're going to finish up today with cell wall structure and start a bit on membranes, then finish mostly cell organelles then in the next lecture or so, and then go into the other topic, which is just a really brief overview of genes and gene regulation. And I want to start back at this slide, and actually let me move back even one more, to talk about cellulose as a beta-1,4 glycoside. So what you see in cellulose is just a chain of glucose molecules strung together. Okay? And if we change beta to an alpha-1,4, then we're looking at starch, not cellulose. So the plant uses then mostly glucose. So if you look at a tree, you're seeing glucose molecules and other components of the cell wall. And you're seeing glucose molecules that are strung together in a beta linkage. So basically, if you were to look at it, every other sugar entity is turned upside down. And then, and we'll talk about starch again, then the plant stores glucose as starch with exactly the same structure, except it's an alpha instead of a beta. So we cannot digest cellulose. We can, of course, digest starch. So it's pretty neat because it uses that repeating unit for two major things in the plant. Then, if we go to the other components that we find in the cell wall, like glycans, they have glucose units. But then they can have, in this case, xylose, which is just another sugar, another 6-carbon compound, that will branch out. And the glycan, and the cellulose glycan then is embedded in pectin. And again, this cartoon is sort of a rough view of what we think, and I've got one, I'll either update you or send you out one of the pictures. So you have the cellulose, which is nothing more than a bunch of glucose molecules in a linear fashion with no groups on the side, so just, beads of glucose. And then the strength comes in because of the interaction of glucose with these other carbohydrates. And I think in this one, pectin is sort of a purple color. And here's a xyloglycan. And so they form bonds. Okay? And they can form covalent bonds. So those cellulose fibrils are tightly held together, and that plant cell can't expand. Okay? Later on, we'll be talking about hormones, and I think I mentioned last lecture, we'll talk about how auxin causes cell walls to elongate. And just to tell you what the take-home message is you can dump a bunch of protons into the cell wall, and you can cause the bonds between the large cellulose microfibrils and the pectins and the xyloglycans to come apart. So by throwing a lot of acid into the cell wall, then the cellulose fibrils start to separate. Now, what's the force that causes them to start separating? What drives cell elongation? I've said it several times. Huh? Auxin, but what causes it to actually, auxin loosens up the cell wall components. But what causes a cell to expand? What's the driving force? Water pressure. Remember, you've got that balloon inside the wooden box. So, in a normal cell, in a plant, at every single moment, there's water that's being pumped into that balloon. And so when the cellulose can start, the fibers can start sliding by each other, it's the pressure, turgor pressure, and you're going to know a lot about turgor pressure, that actually is the force that causes the cell to grow, to expand. Okay? And when I taught the plant physics lab, this is a very neat experiment to do to show that auxin caused cells to elongate. Okay, what's pectin? It's a polysaccharide, used as a gelling agent. I'm not going to go over this a lot. Here's another cartoon, and we're going to go over cell organelles next lecture or whenever. And the question is, how do you make a cell wall? How do you make this massively long chain of glucose molecules? Well, it involves a lot of different organelles. So you've got stuff made in the endoplasmic reticulum, which you've all probably had cell structure before. Then it moves into the Golgi, and then the contents of the Golgi then move out and coalesce with the cell membrane and dump the precursors of some of the molecules then into the cell wall area. Okay? But a lot of the synthesis occurs, as you're going to see, on the membrane. So the question for a long time, and we don't fully, completely know how it works, was how do you make this big, long molecule outside of the cell? That's hard to do. You know, inside the cell, we've got all the enzymes. But how do you make something so gigantic, and you make it a little bit at a time, 1 or 2 glucoses at a time, and what we find is that it actually grows out of the cell membrane. So in this cartoon, you can see these vesicles that are dumping precursors of what they have here, galacturonase, or glatron rather. So these precursors, and you also see these little things right here. And these are actually protein complexes that are the site of actual cellulose synthesis. And one other thing that I wanted to include in here is that calcium is also a really, really incredibly important part of the membrane. Without calcium, the cell walls have no rigidity. Without calcium, what happens is the balloon expands until it explodes. Okay? And you may have seen that stuff on the left there, which is called blossom end rot. And what you're seeing there is a calcium deficiency in the tip of the tomato. Because that, if you do have some deficiencies, that's where it starts to be. So what happens is there's not enough calcium to keep the cell walls together. So you've got the balloon inside expanding, and the cell wall can't contain it, so boom, the cell dies. And so you can see this very characteristic symptoms. What you're seeing there is a calcium deficiency, and what calcium does is it keeps the cell walls together. Okay? That makes sense? You see this a lot, of course, when it's hot or whatever, or there's not enough fertilizer. Different varieties grown at different places can have it. It can be a really major problem. Now, If we go back and talk about how the cellulose is actually made, and that is what we now know is there's an enzyme called cellulose synthase. And if you remember, almost any word that has ASE at the end of it, we're talking about an enzyme. Okay, and an enzyme is a protein that does some reaction, does a single reaction. And believe me, you're going to know a lot about enzymes. And it's a very complex, and we can actually see it. And here is the proteins embedded in the cell membrane. Okay? So inside the cell, we have the precursors, which are sugars attached to UDP, and they enter this complex, and then by moving through the complex, then you have the synthesis. So it's like think about just squeezing a toothpaste tube. Stuff's coming out of the top. If it's cellulose synthase, what would be coming out is cellulose. Okay? Very complex thing. Also, you're going to learn, if you don't know now, that there are a lot of motors inside cells. And I mean things that spin around that have ratchets so they'll have to stop every once in a while. That's how ATP is made. This also has some rotation to it. So every cell you have, you've got a lot of motors. You've got called ATPases that are found in the mitochondria and other places. And they're literally spinning around. And we find that more and more of these things actually move a little bit. And part of that the energy of the movement helps some reactions to occur. Okay? Now we still can't take this thing completely apart. Here's another cartoon showing you basically the same thing. You have this cellulose synthase complex, and in this case you have sucrose donating the glucose, and you can see these chains growing out here. And if you look at this, you can even see, if you take a membrane and split it, which is really not that hard to do, you can actually see most of these things happening. And it's called a rosette because it looks like a rosette. Now this is a newer model of the rosette. And basically this is again just another cartoon that the Golgi produces these secretory vesicles which can go to the cell membrane and put their components out. And at the same time, you have all these little rosettes producing the cellulose. So you have the cellulose being, growing out of the cell membrane, and then the other components, the pectins, the xyloglycans, whatever, are sort of dumped out there and become part of the cell wall. So basically, if you were to be able to stand inside a cell wall, then you'd see these glucose, these cellulose, like rebar, basically. And then you'd see these chains holding them together, and if it's a primary cell wall, you could just sort of walk around in there because a lot of it's either going to be gel or liquid. So it's very strong, but it has a lot of empty space, if you get my meaning. It's not this solid piece of cellulose. Not till later does it become this solid wood. Okay, questions? Okay. Now let's turn it into wood. So we have this primary cell wall that is fluid to a bit. You can squeeze it. If you could take one, you could squeeze it this way. It's not rigid. So it's a very thin cell wall. All cells have primary cell walls. And then later on, when the cell has reached its maturity, whatever that is. So if it's a xylem and it's a long pipe, then in the case of xylem, then it starts producing secondary wall material, and it also produces something called lignin. Okay, and what lignin does is it's the cement. So you go from the pectin gel to a cement. So now you've got rebar and cement. And why do we build things with rebar and cement? Because we have strength, we have some ability to move, but it's not going to start stretching apart or anything. And lignin is what makes wood wood. Okay? We still don't quite know what lignin is. I think I was telling you last lecture that there's a lot of work that's being done on what wood really is. And if you want to make paper, then you want wood without any lignin. If you want to build a house, then you want wood with a lot of lignin. Because lignin gives you the strength. Okay? So we know it's these different kinds of alcohols. And just as a side there, that's what gives the smoke the flavor. So there's some wood you use to smoke. There's some wood you wouldn't use to smoke meat with. Lignin content also can determine at what point the wood burns. So people use mesquite. Mesquite's great to use if you're cooking something like a steak, because mesquite burns hot. A lot of people use mesquite flavor. There's really not much flavor in mesquite. There's a little bit. So usually you're using hickory, pecan, these kind of woods that the smoke has a flavor that's going to taste good or smell good. You don't actually taste smoke. You actually smell it when you're eating it. And most of the flavor compounds are part of this lignin complex. Okay? And what you see in this cartoon is that you have all of these massive number of bonds being formed. Once that lignin is there, that cellulose is never going to stretch. You can't unlignify something. It's a one-way reaction. Okay? And that's because you don't want to maintain it. If it's xylem, and xylem is dead at maturity, you start building up the secondary cell walls, then the cytoplasm goes away, and you're left with the hollow tube to move water up into the plant. But you don't have to worry about maintaining the lignin. Once it's done, it's done. That cellulose is never going to move. Okay? Questions? Okay, now we'll go right back into the next thing inside the cell. So if we start at the outside, we have the middle lamella, which is the stuff between the two cell walls, primary cell walls. Then we have the cell wall, and then the next thing is the membrane. Okay, and like I told you last time, if this is a cell, if this room is equivalent to a plant cell, you'd barely be able to see the membrane. It'd be so thin. But think how strong that is. how incredibly strong it is. And membranes are very, very strong. You could put your hand through a membrane, if it was, this big or so, and you could just move your hand around and really mix it up, pull your hand out, and it'd go right back to the way it was. Okay? Now, let's talk about the kinds of bonds. And everybody knows that a hydrogen bond is a very, very weak bond. Right? Well, it's weak in terms of the chemistry of bonds. And what we determine is how much energy is required to break the bond. So if it's a covalent bond, you've got to pull those atoms apart, okay? Hydrogen bonding is an extremely strong bond. You've probably all seen pictures of astronauts trying to drink water on the shuttle. And the water forms a sphere because water hydrogen bonds to itself. And in fact, if I had a bucket of water on the space station, I could put my hand in that bucket and I could pull the water out. And the water would form a sphere around my hand. So yeah, it's a weak chemical bond, but it's incredibly strong if you've got gazillions of them. And we really depend on hydrogen bonding for most of what we do. Okay? And you all know that the hydrogen bond is due to the polarity where you have electrons that are not equally shared among all of the atoms. In this case, there's, of course, water. So oxygen is going to be relatively negative, and the hydrogens are going to be relatively positive, and so they form these bonds. Now, what is something that is completely in solution? What's the chemistry of that? This. You have water surrounding individual molecules. Okay? That is something, as opposed to shaking up oil and vinegar. That's an emulsion, right? They're not really soluble. Usually it's compounds that are able to form hydrogen bonds, and for something to be completely solubilized, if it's in water, every molecule will be surrounded by water molecules. And that's temperature dependent. Also the negativity of the compound you're trying to solubilize. Now, if I go too well. Now, let me go over this a little bit, because this is why membranes are so incredibly stable. Membranes are composed of polar and nonpolar parts. Okay, and I'm going to tell you the take-home message, and then I'll probably tell you the take-home message again and again. And if we look at this cartoon, on the left we have a typical generic membrane or lipid molecule. You have a chain of carbons. So is that chain of carbons polar or nonpolar? Nonpolar. Basically, you just got a chain of carbons. But then at the head end, the top end, you have attached to it a hydrophilic group. So you have an area that is polar and an area that is nonpolar. Okay? So here's the beauty of it. If we put these in a solution, what they will do is form this structure called a micelle. Here's one, where the polar parts are sticking out into the water, and the nonpolar parts are as far away from the water as they can get. Does that make sense? So they're going to form a micelle, a sphere, with the polar heads soluble in the water and the lipids as far away from the water as that they can get, if that makes sense. Okay? So you shake this up, you shake it up, it's always going to go back to this micelle. It's chemistry, it's basic physics. And I use a lot of theological talk, like, the lipid really doesn't try to get away from the water. It's trying to get bonds with other nonpolar compounds, and water is going to stay on the outside. Okay? So it's very stable. Now the take-home message is that membranes are composed of 2 layers of these compounds, which is why they are so incredibly stable. And, so if we look at this cartoon, we have, in this cartoon, we have the outside of the cell and the inside of the cell. Okay? And it's composed of 2 halves. So you have this lipid, this phospholipid, and then you have another layer here on the bottom. They are situated so that the polar regions are sticking out into the water and the hydrophobic regions are away from the water. Okay? Does that make sense? Now, there are other things. There are proteins. There are different kinds of proteins, and we'll get to those in a minute. There can be other additions. There can be carbohydrates. There's a tremendous number of proteins in the membrane. And there could be also, I think I said carbohydrates. So if we look at this, all membranes have this lipid bilayer. Okay? And we can change the function of the membrane by putting different proteins in it. So basically, all membranes are the same. And there can be different lengths of the nonpolar chains, and it's the proteins that go in them that make them unique. So a chloroplast membrane has chlorophyll embedded in it, and it has enzymes that are required for photosynthesis. Mitochondrial membranes have enzymes that make the mitochondria work. And I told you before, if you took this, let me go back. Well, just as that, you can put your hand through this. If you could blow a membrane up, it's liquid. It's fluid. And you could stick your hand through it. You could pull, you could start just scooping up lipids or phospholipids, and they would all go back to exactly the same way they were, because thermodynamically it's the most stable structure it can form. You're never going to see a membrane with the lipids on the outside. That's just not going to happen. They're not water soluble. They don't like water. So you have this very thin but incredibly powerful membrane. If the cell is the size of this room, you'd still be, the cell membrane would be less than the thickness of saran wrap. Okay? It's remarkable. Very simple. Membranes are not going to turn inside out. The only way you could get a membrane to turn inside out would be to Maybe put them in a lipid solution instead of water, put them in lipids. Maybe that would happen. I don't know. That might actually flip them. That'd be a good test question, actually. Or take-home question. But anyway, they're very stable. We can pop membranes in and out. Proteins in and out. And let me go on down here. Basically, don't worry about the structures. All you should know is there's a hydrophilic and a hydrophobic region, and that we can change these molecules to change the kind of phospholipids. And I went through that. Let's go through now the kind of membrane proteins that we find. The first one is called an integral membrane protein. And in the case of an integral membrane protein, it goes across the membrane. So it has a piece sticking out of the membrane and then it has a piece sticking inside the cell. Okay? We have a lot of integral membrane proteins that transfer molecules in and out of cells, transfer protons. We have other kinds of proteins called peripheral proteins. And peripheral proteins are on one side of the membrane or the other. Okay? So the cellulose synthase complex is a complex of integral membrane proteins because they go through the membrane. Glucose goes in the bottom, cellulose comes out of the top. Okay? Actually, just let me stop. Well, let me just give you an aside here. We can actually use these. This is some of the functions of these proteins. You probably have all heard of experiments or treatments that are being developed where we know when a cell becomes cancerous that it actually changes the membranes in that cell. And so there's a lot of work identifying these unique cancer proteins. And there's been some success, not a lot, but some success in you can make an antibody to that specific membrane protein. So this protein will only be found in a cell that's going crazy, that's become cancerous. And then you can attach to that antibody a poison, even radiation, something that's radioactive, and give it to the person, and it's only going to concentrate, it's only going to bind to cells that are cancerous. So you can give someone a non-lethal dose of whatever radioactive isotope it is, and the notion is it's going to concentrate around those cancerous cells and kill them. So because, it's radiation. It's going to produce usually not gammas, betas, whatever. And it's only going to concentrate to a lethal dose in those cells that are cancerous. I had a technician years ago who had a really strange kind of cancer called carcinoid syndrome. And they were using this treatment. She had to they only did it at LSU. So she would go to LSU like once every 2 months or whatever and get the treatment. And she had to stay there for several days. She went once and got the treatment, and there was a hurricane coming in, so she had to come back. And just walking down the hall, she pegged the Geiger counter in the lab. And the treatment was to give her a lower dose than a non-lethal dose and count on the radiation becoming at a lethal level where the cells are. And there's all sorts of work going on about that now. Now, There's different kinds of proteins and different ways that things get into cells. On the left-hand side, we have something called just diffusion. That is, a molecule just can go across the cell membrane. Very few things actually diffuse into the cells. There's even proteins that carry water into the cell. Most everything gets into the cell through proteins. And you can have a channel protein. What's a channel protein? If you look at it, and I'll have some, I think probably will go in the next lecture, it can just pull the molecule through the center of it. It's basically an opening. Okay? You can also have a different class of protein called a carrier protein. What's the difference between a channel protein and a carrier protein? A channel protein, the molecule moves through an opening, like a pore. A carrier protein grabs the molecule and pulls it through. Okay? So it literally binds to the molecule and pushes it to the other side. And these are all that I've talked about so far are passive transport. Okay? The one that actually is probably the most used is the one called primary active transport. Okay? And that requires energy, almost always ATP. And these active transport mechanisms, this is how almost all minerals get into plants. And they can go inside or they can go outside. You can pump things into the cell. You can pump things out of the cell. You can pump things into the mitochondria with a transport protein that's in the mitochondria. And cells have hundreds of these, different kinds. Almost all minerals get into the plant by active transport. Very few just diffuse through. And you'll see that when we talk about mineral nutrition. You'll see this again, like everything in here, you see it again, again, and again. Also, this active transport, another factor is that it can work against the concentration gradient. Questions? Okay. Let's see if this is... Okay, I'm going to send this out for you, or I'll make a video. It's just, for some reason, it never will open here. So, well, I will see you on Wednesday. Also, there's a discussion page. If you have, you can email me, or you can put up a discussion question if you have something needs to be discussed. And I'll check to be sure all the lectures are up. Why does this never work? It never has worked down here.